Namaskar. Today we are here to discuss about bacterial photosynthesis, its light reaction. Me, Neela Upadhyay, and my colleague Gayatri Nagar would discuss about bacterial photosynthesis. We have decided to discuss three main parts of this topic, that is Anoxygenic Photosynthesis, Oxygenic Photosynthesis and Rhodopsin-Based Phototrophy. Photosynthesis is a very significant process. It is, we can say, the most significant process on earth, as sun is the main source of energy for life on earth, and we are dependent on this energy. This is the photosynthesis process which can transform solar energy or light energy to that of chemical bond energy, and that is why this process becomes very, very important in nature. First, let us understand what is photosynthesis. There are two principal reactions in photosynthesis, that is light reaction and dark reaction. During light reaction of photosynthesis, ATP, reducing power, and oxygen are generated. And during dark reaction, ATP and reducing power is utilized for synthesis of complex carbohydrates. Overall reaction of photosynthesis in bacteria is carbon dioxide plus H2A that produces CH2O, that is carbohydrate. So CO2 is oxidant and H2A is reductant. That is the case with purple and green bacteria. Another equation shows the photosynthetic reaction of cyanobacteria, where carbon dioxide is electron acceptor, H2O is electron donor, and ultimately carbohydrates are produced, that is CH2O, and during that oxygen is evolved. Photosynthetic process uses light energy. Light energy comes as a discrete packets which are named as photons. These photons are accepted by the pigment molecule, and due to that normal pigment molecule changes. Redistribution of electron, as it is shown in the figure, takes place. Redistribution of electrons results in synthesis of high energy state. So pigment molecule comes to its high energy state, that is the excited state. Excited state can release the electrons, and these electrons which are released from excited state, they are accepted by electron acceptors, and due to that reduced compound is synthesized. The excited state pigment molecule, after losing electron, now becomes oxidized and it is ready to absorb more photons. So this is the basic principle which is applied in the photosynthetic process. Photosynthesis is carried out in number of bacteria, and they are various groups of photosynthetic bacteria are pulper bacteria, which includes purple sulfur and purple non-sulfur bacteria. Example of purple sulfur bacteria, that is chromatium. Example of purple non-sulfur bacterium, that is Rhodobacter, Rhodopseudomonas, Rhodospirillum. Green bacteria is also an important group. As the example, green bacteria also incorporates green sulfur bacteria and green non-sulfur bacteria. Chlorobium is the example of green sulfur bacteria. and Chloroflexus is the example of green non-sulfur bacteria. Then Heliobacteria, they are the only gram-positive organisms which can photosynthesize. Helobacillus, Heliococcus, these are the representative genera. Cyanobacteria is a very important group because they carry out oxygenic photosynthesis, they evolve oxygen during the photosynthetic process. And last is Halobacteria, that is entirely distinct, and that will be explained by my colleague. It is considered under Rhodopsin-Based Phototrophy. These are the shapes of different bacteria which are capable of photosynthesis. Figures of purple sulfur, purple non-sulfur, green bacteria, Chloroflexus, Heliobacter have been given. The question is where photosynthesis is carried out. Photosynthetic apparatus is there in all the systems, and in the photosynthetic apparatus, photosynthetic pigment is present. The photosynthetic apparatus is organized in a different way in different systems. It may be in a form of extensive intracellular membrane system, which may be vesicular, lamellar or tubular. This is the characteristic of purple or green bacteria. Chlorosome. Chlorosomes are special structures which are surrounded by non-unit membrane, and these chlorosomes, in the chlorosomes photosynthetic pigment is present which carries out photosynthesis. And third one is chloroplast. Chloroplast is a characteristic of cyanobacteria. Chloroplasts are consisting of thylakoids. Stacks of thylakoids makes grana, and thylakoid membrane is attached to phycobilisomes. So there photosynthetic pigment is located. These are the structures of internal membranes of photosynthetic bacteria, and the structures are in a form of tubules, vesicular, then stacks, lamellar, etc. This is the slide which shows chlorosomes. Chlorosomes are found in green bacteria. The thing which is shown in green color, it carries cigar-shaped object and which carries bacterial chlorophyll. Beneath it, membrane is shown. On the right hand side, membrane proteins, red colored membrane proteins. Now brown colored things are showing reaction center. Yellow colored is light harvesting complexes, and next to it, that is ATPase. So bacterial chlorophyll, which is present in chlorosomes, first absorbs the light via light harvesting complex. It is transmitted to reaction center, and then the process of photosynthesis starts. These are the components of photosynthetic apparatus. We have discussed about photosynthetic apparatus, types of photosynthetic apparatus. Now this apparatus are made up of antenna of light harvesting pigments, photosynthetic reaction center, and photosynthetic electron transport chain. We will discuss about each component of photosynthetic apparatus. First is antenna of light harvesting pigments. In antenna of light harvesting pigments, main pigment is chlorophyll. Structure of chlorophyll is shown. It is very much similar to that of cytochromes. In cytochromes, iron is present in the integral part. Here magnesium is there. Tetrapyrrole ring structure is present as it is found in the cytochromes. In chlorophyll, additional fifth ring is present, which is not present in cytochromes. Major chlorophyll which can carry out photosynthesis process are chlorophyll A and bacterial chlorophyll A. Structures are shown. Only the side chains differ in bacterial chlorophyll A, otherwise the things are same. Now you can see phytol as the side chain, which is again not present in the cytochromes. Carotenoids are also present in the antenna of light harvesting pigments. Now these carotenoids are of different types. Few types have been shown. Carotenoids have two principal functions. They absorb the light having 450 to 550 nanometers, and that way they help in the conservation of light energy. And another important function of carotenoids, that is, they protect photosynthetic apparatus from photooxidative damage. This is photosynthetic light harvesting. I told you about antenna of light harvesting pigments. Now this is the antenna of light harvesting pigments. On one side you can see photons which are absorbed by the molecules of antenna, and because of the absorption of light they become excited and they will donate photons to the neighboring molecule. Ultimately they reach to the photosynthetic reaction center. In photosynthetic reaction center also chlorophyll and many other pigments and accessory pigments are present. So ultimately the photons, that is which gives light energy, that is transmitted to photosynthetic reaction center, and the way of transmission, that is by inductive resonance. This is photosynthetic reaction center and light harvesting complex. Now the A and B both are crystallized structures of this light harvesting complex and reaction center. This different colored, yellow, blue and red colored, this shows different polypeptides which are associated with the reaction center and light harvesting complex. Now we actually discuss anoxygenic photosynthesis. Anoxygenic photosynthesis is carried out in mainly purple bacteria, green bacteria and heliobacteria. They all differ in the basic components. Reaction center chlorophyll is bacterial chlorophyll A in purple bacteria, bacterial chlorophyll A in green bacteria and bacterial chlorophyll G, that is in heliobacteria. And similarly there is difference in antenna accessory pigments. Now source of reducing power differs in all the bacteria. In purple and green bacteria it carries H2, H2S, reduced organic compounds, but in heliobacteria lactic acid and certain organic compounds act as the reducing power. And the source of carbon, that is carbon dioxide or organic molecules in purple and green bacteria, and in heliobacteria they are organic molecules like lactate and pyruvate. Anoxygenic photosynthesis has been studied in detail in different bacteria. This is the comparison of purple bacteria, green bacteria and heliobacteria. We can see that the reaction center, only one photosystem is present. You will see in cyanobacteria that there are two photosystems. This one photosystem absorbs the light of 870 nanometers. In case of purple bacteria, in green sulfur bacteria 840 nanometers, and in heliobacteria 798 nanometers. Now in all the bacteria you can see cyclic flow of electrons, that is P870 absorbs the light, it becomes excited, it is ready to release electrons, electrons are accepted by bacterial chlorophyll, then bacterial pheophytin, then coenzyme Q to cytochrome B C1, then cytochrome C, and again back to P870. That means flow of electrons is cyclic, chlorophyll to chlorophyll. That is why the phosphorylation which takes place during this purple bacteria, during this process, that is called cyclic photophosphorylation. Actually, ejection of electrons is energy consuming process, but when electrons are transported from one carrier to the other carrier, from negative to positive redox values, at that time there is generation of energy. Now the product of light reaction is also reducing power, as I told you before. So reducing power is generated in a different way. There is existence of reverse electron transport chain in purple bacteria, where from coenzyme Q electrons are accepted by NADH. Actually directly, due to the difference in redox values, coenzyme Q cannot donate electrons to NADH, and that is why reverse flow of electrons is necessary. So what is happening, that from bacteriophyophytin electrons move to coenzyme Q, then cytochrome B C1, and this cytochrome B C1 will accept electrons from electron donors like H2S, elemental sulfur, thiosulfate, hydrogen, etc. And these electrons move in the reverse direction also to reduce NAD. So that way reducing power is generated by reverse electron flow, and normal cyclic pathway gives the synthesis of energy. Similar is the case with green sulfur bacteria. I won't repeat the things, but this is also cyclic flow of electrons. Only the difference is from iron sulfur proteins, electrons can be easily accepted by ferredoxin, and ferredoxin will be reduced, which can be easily used as a reducing power for reduction of carbon dioxide. And similarly in heliobacteria also, from iron sulfur proteins directly electrons can be accepted, and this is again possible because of the specific redox values of this different components. In anoxygenic bacteria, purple bacterium has been studied in much more detail, and this is the light reaction in purple bacteria. Of course, same components we have seen in the schematic representation. We can see light which is absorbed by antenna and light harvesting complex, LH, that is light harvesting complex. From light harvesting complex, as I told you, photons, that is the energy, moves to reaction center, and this reaction center, that is P870, it becomes excited. P870 donates electrons to bacteriophyophytin. From bacteriophyophytin, they reach to coenzyme QH2 to cytochrome BC1, iron sulfur protein C2, and then again back to photosystem. Now here main thing which has to be mentioned, that is like respiratory electron transport chain, there are certain electron carriers and there are certain hydrogen carriers. Quinone is the hydrogen carrier, and cytochromes BC1, they are the electron carriers. So when quinones are donating electrons to cytochrome BC1, at that time protons are spared, because BC1 will only accept electrons. These protons are spared and they move on the outer direction, and they are deposited in a form of proton gradient. These electrons has to enter within the cytoplasm, and structure of ATPase has been shown, via which these protons can come back to the cytoplasm, and so ATPase is activated and it will phosphorylate ADP to produce ATP. So this way we have studied anoxygenic photosynthesis. Now oxygenic photosynthesis will be discussed with by my colleague Dr. Gayatri Nagar.
Hello friends, today I will discuss the oxygenic photosynthesis. Sorry for the interruption. Oxygenic photosynthesis, as it is named, here in this process the oxygen is evolved, so it is called the oxygenic photosynthesis. We discussed the light reaction, in which the radiant light, it is absorbed by the phototrophs, and this light energy, it is converted into the chemical energy, and during this the energy as a ATP and the reducing power NADPH is produced. So in oxygenic photosynthesis, the organisms involved, they are the phototrophic eukaryotes, which includes the plants and the algae, and the prokaryotes, they are the bacteria, cyanobacteria. Cyanobacteria, they are the main microorganisms for the photosynthesis in which the oxygen is evolved. When we consider photosynthesis, most of the people, they think the plants are the main organisms which are involved in the photosynthesis reactions, but it is not true. 50% of the photosynthesis on the earth, it is carried out by the microorganisms, and in which the cyanobacteria, they are the main bacteria which carry out the oxygenic photosynthesis and contributor of the oxygen on the earth. This slide, it shows the images of the cyanobacteria. There are various types. The main light absorbing pigments in the cyanobacteria, they are chlorophyll A, chlorophyll B, carotenoids, phycobiliproteins, phycoerythrin and phycocyanin, in which these pigments, where they are present in the phycobilisomes of the cyanobacteria. This slide, it shows the cellular structure of the cyanobacteria. It shows that they are the prokaryotic cell, where the DNA that is naked, and the phycobilisomes, they are present as a tiny knobs on the lamella. Phycobilisomes, the pigment protein complex involved in the photosynthesis. It is a subcellular body, appears on the tiny knobs attached to the outer surface of the photosynthetic membranes. The light energy, it is absorbed in the phycobilisomes and it is passed to the chlorophyll A, and from there to the reaction center, that is the photosystem 2. This phycobilisomes, they contains the photosynthetic pigments, we call them as a phycobiliproteins. These phycobiliproteins, they are of two types, phycocyanin and phycoerythrin. Collectively they are called the phycobiliproteins. The phycoerythrin, it absorbs the light around the 550 nanometer, and phycocyanin, which is blue in color, it absorbs the light around the 630 nanometer. Due to them, the organism can capture the incident light that falls in the narrow range. These pigments, they are the accessory pigments in the cyanobacteria. The main photosynthetic pigment in the cyanobacteria, they are the chlorophyll A and chlorophyll B. The chlorophyll A pigment, it absorbs the light at a peak that is at a 665 nanometer, and the chlorophyll B, it has the peak at 645 nanometer. Chlorophyll A, it also absorbs the light at 430 nanometer, which is the blue spectra. So this chlorophyll pigments, they absorbs the red and blue light. So when they emit the light, they emit the green light, and so they are green in color. The plant's chlorophyll, it has the similar nature like the cyanobacteria chlorophyll, and so they are also green in color. Other accessory pigments, which we call them as a phycobiliproteins, which are two types, phycocyanin and phycoerythrin. These two are the accessory pigments because they help the cyanobacteria for the photooxidative damage to the cell. When the high intensity light, it is passed through the cell, it doesn't create the oxidative damage. So they are called the accessory pigments. The another role which is played by this phycobiliprotein is it also absorbs the light in the green to yellow range, and so the bacteria has the broader wavelength of the light for the absorption, and this helps the photosynthesis reactions. It also protects the microorganisms from the sunlight damage, which I have already narrated. Now, this cyanobacteria, they are evolved first on the earth 2.5 billion years ago. They have generated oxygen by developing the two independent reactions. We call them as a photosystem 1 and photosystem 2 in the cell. And this two system in the cyanobacteria, they have evolved the oxygen on the earth, and it has created the oxygenic atmosphere. And later on, the aerobic respiration in the bacteria developed, then the archaea and the eukaryotes, they develop. So when we see the evolutionary image on the earth, the cyanobacteria, they are the first bacteria to evolve on the earth 2.5 billion years ago. Now I have told that the two types of the photosystems, they are present in the cyanobacteria. The photosystem 2 and the photosystem 1. The photosystem 1, it absorbs the light greater than the 680 nanometer wavelength, and it is shown that this is the photosystem 1. The light is absorbed by the photosystem 1, and the P700 center of the photosystem 1, which contains the chlorophyll, that is excited and it releases, it is oxidized, and it generates the two electrons. The electron, these two electrons, they are generated to the carriers, and these carriers, they are the ferredoxin. From this, it goes to the FAD, and then the reducing power NADPH, it is produce. So by this way the reducing power NADPH that is produced. So this is a one way of the production of reducing power in the photosystem 1. The another way of the travel of the electrons, that is from the pyridoxine, it travels back to the cytochromes, and these electrons, it travels to the cytochrome B and then to the plastoquinone and then cytochrome B6, FES, cytochrome F, and then these electrons, they are donated again to the P700, and thus the P700 that is reduced and ready to accept the other light. During this, it is a cyclic photophosphorylation where the ATP is generated when the electron, it passes from cytochrome B to the plastoquinone. The ATP is generated, and so this type of photophosphorylation we call them as a cyclic photophosphorylation. So that is about the photosystem 1. Now the role of the photosystem 2 in the cyanobacteria. The photosystem 2 reaction center, they absorb the light at lesser than 680 nanometer, and the peak for the chlorophyll, that is the reaction center, that is P68. P68, that is the light absorbing center for the photosystem 1. As it absorbs the light, it is excited, it is oxidized, and it releases the electrons. These electrons from the photosystem 2, they are then transferred to the various cytochromes and to the phaeophytin, and then this way it is transferred to the various plastoquinone, and then ultimately the hydrolysis of the water occurs and the oxygen that is evolved. So it is a way of the non-cyclic type of the photophosphorylation. Now the second is the photolysis of the water in the light reaction. The photolysis of the water that occurs in the photosystem 2 of the cyanobacteria. Photosystem 2, it has the enzyme, we call them as a S system, and this enzyme, when it is in the ground state, it is the S0. This enzyme, it couples with the two molecules of the water, and then it absorbs the light and it is converted into the S1 stage, excited, again it absorbs the photon and converted into the S2 stage, again it absorbs the light and converted into the S3 stage, and the most excited stage, that is the S4 stage of the enzyme S system for the photolysis. During this, the water molecule that is not hydrolyzed. When the enzyme that is under highly excited stage, that is at a S4 stage, the electrons, they are released and the photolysis of the water occurs. This occurs, oxygen is released, 4 hydrogens they are produced, and the enzyme that again come back to the ground stage. That is the photolysis in the cyanobacteria. Now, the another type of the phototrophy, that is rhodopsin-based phototrophies, and this is found in the halobacteria. The halobacterium, halobium, they are the bacteria which is the light absorbing pigment rhodopsin. This rhodopsin, it is the pigment which is purple in color, and it resembles with the retinal pigment of the vertebrate eyes. This pigment that is responsible for the phototrophy in the halobacterium, halobium. The pigment absorbs the light and it undergoes the conformational change, and it takes the light energy and it transfers the hydrogen ion protons to the periplasmic layer. So, it creates the proton motive force. So, by this way, the bacteriorhodopsin which is present here will transfer the protons to the periplasmic layer, and it produces the ATP in the bacteria. The proton motive force that is responsible for the generation of the ATP, because these protons, they produce the pH gradient, and this pH gradient, it causes the production of the ATP in halobacterium, halobium. This halobacteria, they are obligatory aerobic, and they operates the membrane bound respiratory pathway for the ATP generation. As they are aerobic, they use the organic carbon as a source of energy, but during the low oxygen content, this bacteria, they use the bacteriorhodopsin. During the low oxygen content, this bacteria which are present in the marine sediments, a high salt environment, they synthesize the bacteriorhodopsin in their cell as a purple patches, and this bacteriorhodopsin, it absorbs the light, and this light energy, it creates the proton motive force and generates the protons in the periplasmic layer, which enables the synthesis of the ATP. This figure, it shows the habitat of the halobacteria in the Red Sea. These are the diagram of the halobacterium, halobium. The rhodopsin, the proton motive force. The quantum requirement for the ejection of the proton by the bacteria, that is approximately 2. When we compare them with the purple photosynthetic bacteria, it is only 0.5. So, the efficiency of the photosynthesis, that is only 25%, but when it is used by the bacteria, when the oxygen content in the saline water, it is very low, and under that condition, as the bacteria are aerobic, they can survive by this rhodopsin-based phototrophy, because due to the presence of this pigment, they can absorb the light energy and convert them into the chemical energy as a ATP. But the main source of the energy, that is the carbon source, organic carbon. The recent studies in halobacterium, there are bacterial species in the marine sediments in deep sea that absorbs the blue light which penetrates to the deep sea sediments. Their rhodopsin, it differs with the proteorhodopsin. A number of proteorhodopsin variants, they have been found, and it represent the recent biodiversity of the habitats, and they probably support the significant biomass in the environment. So now Neela madam will explain you the significance of the photosynthesis.
We have discussed about anoxygenic, oxygenic photosynthesis and rhodopsin-based phototrophy. On the basis of that, in nature, photosynthesis process becomes very important. It can be considered as most important process. Different points are: phototrophs reduce and incorporate carbon dioxide. This is the only mechanism by which carbon dioxide can be fixed. So that way atmosphere will become more and more clean.
Video आप तो मैं
કેમ પેલી સ્લાઇડ કેમ બંધ કરી દે?
ક્લિક કરો પણ આવતું નથી.
સિગ્નિફિકન્સ આગલી સ્લાઇડ. So photosynthetic process becomes very significant metabolic process, as all energy derived from solar energy. It produces organic compounds required for growth. These organisms serves as the base for food chains in biosphere. Cyanobacteria, oxygenic phototrophic organisms, produce oxygen. So oxygenic atmosphere may have been created by these bacteria, and over half the photosynthesis on earth is carried out only by microbes. So thank you. We will see the animation.
Video clipping of the photosynthesis, which you will like.
Very slow.
It is in the thylakoid membrane, and see how the protons they are generated, and the oxygen is evolved, the NADPH is produced, the ADP and PI, this enzyme that produces the ATP. The light energy, it is shown in the arrow, the sunlight energy that is absorbed by the green pigment chlorophyll, and see the protons are released, the electrons from the chlorophyll they are donated. Thank you very much for the listening. If you have any questions, you can always ask to the teachers. I hope you enjoyed the session, and this is covered in the TYBSc syllabus, so it will help you in the exam. Thank you very much.